So again, I'm Dr. Ferguson. The whole goal of this workshop is to get you comfortable using Excel. It takes a while to become an expert in it, but it's a tool just like any of the other tools you've used. Most of you are pretty comfortable with like your TI-86 calculator or whatever it is that you've been using in classes up to this point. You want to think of Excel as the most fabulous and useful calculator you've ever worked with. It's a fantastic resource for you to be able to do data transformations, to present data in a way that's going to be accessible to anybody that you're trying to communicate with. So I want you to be at least comfortable with the basics, and then I'm going to show you some real fancy stuff that you can do with it if you become an expert. So I made a Google Drive. If you're not familiar with Google Drive, and that's not really the focus of this workshop, but I want to bring it up so that you know that this is another way that you can collaborate between each other. So as part of our Google suite of tools that we have at Fredonia, if you go into your email, you click this little grid here, and there's this thing called Drive. And so if you come over here to Drive, I've got this folder here. I'm going to share this folder with everybody in the room. So what I'd like you to do is just email me when the workshop is. Rebecca will give me all your email addresses, and I'll share this folder with you. And so you can just get in there and pull down most of the examples I'm going to show you, so that it's not like, oh, how did he do that? What did he say? You can actually go and see the formulas and all the things that I've shown you tonight. So I'm going to open this up here. This is the desktop. I have 2 slides. What you need to know to make Excel work for you, and the little funny bylines, just my musings on Excel. I made a list of topics that I want to show you, but if there's anything that I'm not showing you that you want me to dive into, I'm pretty good at Excel, so I'll try to show you whatever I can. Come on. There we go. Oh, it was you.
Oh,
you were
sorry.
coming down the hall. It was communicating with my.
Oh, here you go.
There we go. Thank you. There we go. So the thing that most people know when they open up Excel is there's this whole grid of boxes. And unfortunately, for a really long time, most students think that's all it is. It's a place to organize your data, but not to do any analysis or manipulation with it. Entering data into the spreadsheet is just the very earliest, most fundamental thing that you can do with Excel. But even in that basic feature, formatting your data in an accessible way is an important skill to learn. And then, as important as entering it in is preparing it for subsequent analyses. So you want to have clear headings of each of the columns and rows so that you know what the data are. And then I'm going to teach you a little bit about something called conditional formatting. It was something I did sort of off the cuff when I was talking to those students that prompted this workshop in the fall. I did a conditional format that scaled the color of the cell based on the value that was in it. And they looked at me like, what'd you just do? And it's a real basic way to make your data easily accessible. You can look at it and immediately know, is this a high value, is it a low value? How does it relate to the other values in that field? Probably the most important thing that I want you to take away from this is that Excel excels at performing calculations. Computers are really good at making calculations, but they're only as smart as the formulas that you put into them. I don't know how many of you have run into this, but you'll enter a bunch of calculations into your calculator, and then all of a sudden you get an answer and you're pretty confident that you've typed them into your calculator correctly, but then the answer is wrong and you're wondering why. Oftentimes this is a rounding error. So if you do each step in a long calculation and you start truncating those numbers, like you're going to round it to 1, 2, or sometimes even 3 decimals, you can have a wildly different answer than somebody who uses all the numbers that come out of the calculator. Excel will allow you to take a number that you start with, perform a number of calculations in between, and give you the end result, which is where you should worry about significant figures. When you're having all these intermediate values, you should keep all of the numbers that make up that intermediate value. So by doing these calculations in Excel, it'll protect you from rounding errors. It also allows you to dynamically update your solution. And by dynamically update, I mean you've got value 1, you're going to transform it this way, you're going to do it this, and this. And if you realize you made a step at one intermediate, all you have to do is go back, fix that intermediate, and everything else updates and gives you the answer rather than having to start from scratch and work your way all the way back through. 5 rounding errors twice. The next big thing is graphs. Oftentimes you're asked to prepare graphs for lab reports or different analyses you're going to do. Excel's great for that. There's specific customized statistical software packages. If you take some upper level classes, you might learn SPSS. I know Ramsey's is in biostats right now. But for sort of all-purpose graphing, Excel does pretty well. It can make a whole bunch of different sorts of graphs, and certainly as a place to learn how to format your data in a way that's going to make a graph that communicates your science well to the reader, Excel's great for that. And then it can also make logical decisions for you based on what's called a Boolean operator, and some of you will know what that means and some don't. Boolean logic is if-then statements. So if the sky is blue, then that means that light scatters in a certain way. Equals, greater than, less than. It can make decisions based on the way two cells relate to each other, and it'll do one or the other calculation. It can sort data based on different values. So if you have a whole big table, and I'm going to bring up an example here, with 10, 20, 100 different parameters. And so for a given sample, you've analyzed it for all these different parameters. You can sort based on any of those parameters and see how they relate to one another. I use that all the time. It's a great feature. It's really highlight, click, one step, and you can sort your data in a really easy and effective way. And then, let's see what else I'm going to say. It automatically treats data differently based on the conditions that you specified. And so this logic function, we won't get into that as much, but if you have questions about it, we can do that. So I'm going to open up a couple example files that I have. I'm just going to switch to this, because I have it all organized in a folder here. Excel workshop. Okay, so I'm going to start with, oh, it's not here. Here it is. The fanciest example. It's a spreadsheet that I made as a graduate student, not to do any kind of science. But if you've ever heard of the game roulette, so roulette is a wheel, it's a gambling game, and so there's 38 different spots on the wheel. You put a marble in it, and you spin the wheel, and it goes around that's in the table. And depending on where the marble lands, you win or lose, there's different payouts depending on the bets that you make. And so I made this spreadsheet, sort of a challenge that my brother-in-law made with me. And he says, you couldn't simulate how to do this betting structure that I have in Excel. I said, I think I could probably pull it off. And so over the span of a couple weeks in the evenings, I put this thing together. And the reason I'm showing this to you is because it highlights just one use of Excel. You can simulate a lot of different things that are happening in real life using this tool. So if we go in here, and here's data. On the leftmost column here, I have the result when you spin the wheel. Excel will generate a random number, somewhere between 1 and 38, because there's 38 positions on the wheel, and report that number. Now, a 31 is black on the wheel, so there's black and red spots. It's an odd number, and it's also the higher half of the two. So these are different kinds of bets that you make in roulette. And then in here, this is a part of a sheet that bets when my brother-in-law says you should bet to win at roulette. I said, you shouldn't do that. It's not a winning strategy. And so if we go over here to formulas, and I calculate this, what's going to happen is it's going to simulate 5,000 new spins on a roulette wheel. It's going to determine, is that a red, a black, and there's two spots, the green spots in the wheel. That's why the house always wins. It's because the green spots don't pay out unless you bet on those. And it tells me when I'm going to bet. So here, I placed a $10 bet against the next number being, in this case, even. So there's a whole bunch of odds, and my brother-in-law says you should bet against the run. The reason why this is a horrible strategy is sort of beyond this, but he says place a bet. It came up even, and so that means he won the bet. In your bankroll here, it adds that $10. It pays 2 to 1, so it gives you twice the money back, and you've added $20 to your bankroll. Okay, great. Over the course of 5,000 spins, this is plotting the results of his betting strategy for a given set of calculations. Calculate again. What it's going to do is simulate a whole new set of spins. This graph will update in real time. There's another graph down here. Calculate that. It shows a histogram representing all the different outcomes across 5,000 spins, how many occur. If you know any stats, those of you that have taken genetics, you know about chi-square analysis. So chi-square tells you for a given distribution, if you have a predicted outcome, how does this actual outcome compare to the prediction? And so this number here basically says that 7% of outcomes would be a worse fit to an idealized outcome. And so that's somewhat less probable. If we spin it again, it'll reanalyze it. So that's a slightly better fit. Do it again, it's an even better fit. And so this is really just a quick chance to show off what Excel could do. You are not going to be doing anything this sophisticated probably in your introductory labs or even in the first couple of years. But you'll see all that formatting is changing. The decisions about whether he should be betting or not are changing in response to values that are coming up in that random number generator. So Excel is really powerful. It can do all kinds of fun stuff, like settle bets with your brother-in-law. So I'm not going to save that. But let's start with something a little more basic. So this is a worksheet that I put together here with a bunch of terms that I want to define for you guys and sort of approaches to working with Excel. If we start with sort of basic data entry and organization, because that's what you're going to do. You're going to be in a lab. You'll have an independent variable that you're controlling. And so let's say you're changing concentration of some analyte. And then you're measuring the dependent variable, the response to that initial change that you're controlling in the independent variable and seeing how it responds. So let's say you're adding something to a cuvette and looking at how much absorbance you get in the spectrophotometer. You're going out and you're measuring trees to figure out what the diameter of them are at this height. You're throwing, Dr. Titus has those 1 meter square plots. You throw that thing out and you count how many different kinds of organisms are in that plot. And so what you need to do is organize that stuff in a way that's useful to you. I'm going to toggle back and forth between this somewhat more complete worksheet and something that's totally fresh. And this is the first thing that I want to show you is that this whole thing is called a worksheet. This is a tab at the bottom. And then if you press this plus, it creates a whole new worksheet. Okay? It's not a workbook, it's the worksheet. It's the one spreadsheet inside this file. And you can toggle back and forth between these two worksheets as a way to organize data inside a single file. You'll note that the file name stays the same at the top. So when we go to sheet 2, we can move data back and forth between those two while keeping it organized in the same file. Please interrupt me at any time that we're going along here. So this merge and center, auto width, clear versus delete, and insert, this is just how we're going to move data along. So let's say you have 6 plots. So this is a Dr. Titus style experiment where you go out, plunk some square meter things out in the yard, and you count how many things are in there. Most of you have done that lab as part of the intro to ecology and evolution. So we're going to put that right here. Okay, You've got a heading for that term that you're using in the spreadsheet, and this describes the independent variable, right? You're going from one plot to the next plot to another plot. Okay. Now we're going to say in that plot we saw a couple different things. We saw dandelions and earthworms and clover and periwinkle, so I'm just going to highlight a couple different things here. They're completely fictitious and made up. I sat around last night trying to figure out what I was going to put in here. But we're going to make these different headings. What you'll see is that there's some spacing between them, the way I pasted this in here. Being able to insert and delete rows and columns is something that you're going to want to be able to do effectively. If you put information in a column and all of a sudden you realize you want to calculate some sort of an intermediate manipulation, you want to be able to insert a new column at an appropriate place in your spreadsheet. So I'm going to highlight these two columns here, E and F. Each cell is described by its position in the spreadsheet. So if I click, say, on dandelion, you see it's D2. So it's column D, row 2. But I'm going to highlight both E and F. And I'm clicking on the numbers up here instead of down here. This is different. I'm not highlighting the entire row or column if I do this. You have to click up here. And that goes from row 1 to infinity. There's as many cells as you could ever want in an Excel spreadsheet. It adds more as you add more data. You right-click. That brings up these context-sensitive menus. There's lots of good stuff in here. But what we're going to do is delete these two columns. So if I do that, what it does is it deletes those and it slides everything back over so that they're next to one another. So I'm going to tighten up all these columns here by highlighting and deleting these intervening
I have a
terms.
question.
Go ahead, Ramses.
So, I know the answer to this, but when you delete these columns or row, does it update all the relative positions in the formula?
It does. And that's useful. So what he's asking is if we had a formula here, let me go back. So if we had a formula here that was referring over to this cell, that reference is going to stay constant. And we're going to get into, when we make formulas, what it means to have a reference from one cell to the next, because that's really the heart of how formulas work in Excel. So let's delete these. Delete. Delete. Okay, so I'm going to go back here and copy some phony data. So this is the number of dandelions that I saw. This is the number of earthworms I saw. This is the number of clover plants I saw. Periwinkle wasn't too common in this specific case. And saw a couple honeybees. Okay, so one thing that I want you to notice is earthworm doesn't quite fit into that cell, right? See how it's overwritten by clover? And if this is too basic, please let me know. But what you can do is hover over the division between these two columns. You can double click it. It automatically adjusts the width to how wide that column is. So we can go along here and say, okay, well, let's resize each of these columns appropriate for the contents that are in there. So these are our plots. These are our different things, and this is the number of observations we made in there. Now, one thing that you might want to do is say, well, what's the average number of dandelions that are in there? So we'll do one of our first formulas here. So we're going to type in average, because this is a calculation we're going to do down here. And in this cell, when you're going to start adding formulas, there's two ways to do it. If you know the formula, you just type equals. All formulas begin with equals. So you type equal, and then you type the name of the formula you want to do. So let's do average. So you do average. It comes up with this little shortcut. There's a couple different versions of average. We'll just do the basic one. I know that the way this works is that you make a set of brackets and you can highlight that data. You'll see it says D3 colon D8. That's what you call a range. And so it's in column D, cells 3 through 8, inclusive. Hey, Geraldo. And then you press enter, boom, I've got an average. Oh, I forgot to count this dandelion in plot 3. So I'm going to change that. There's 9 there, actually. I hit that. You see how the average updates in real time? And so you can go back and manipulate any of the intermediates, and it's going to update the final result. The next thing I'm going to show you is I not only want dandelion averages, but I want averages for earthworms, clover, periwinkle, and honeybee. You can type equals average range and do it over again. But if you see this little box that's down here at the corner of that cell, if you hover over it, you'll see that the cursor turns to a little x. So there's the big fat X that you usually see, but then it goes to a little x. This is a formula copying feature. You're going to click and drag. And what it's going to do is calculate the average in that cell of the cells that are in a relative position. And I guess that's sort of a hard thing to explain. Here it says average D3 through D8. So we're going to do average of the cells that are immediately above this cell. But when I drag it over here, you'll note the number changes. That's because in this cell, instead of D3 through D8, it's now E3 through E8. What it's doing is saying, I'm taking this formula and finding the average of the cells that are in the same relative to position as the first formula that I used. And so I can drag it across and it'll calculate the average for each of these. Now, it'll use as many significant figures as you let it. So if you click on that, there's this number here. So there's the number region in this ribbon. And you say, I want more significant figures. I don't believe it repeats forever. It actually does, but uh-oh. That's nothing you need to worry about. That's just my mouse. So you can use more and more significant figures if you want to, but in most cases you're going to just need a handful. And that depends on your observation. If you're using an instrument that only reads out to hundredths of a unit, you don't want to use more significant figures at the end there. You can control how many you're going to use by highlighting this range and increasing or decreasing the number of those significant figures. If you want to go all the way down to an integer, you can do that. But let's say we want to keep 3 sig figs there. So now we've just done our first formula. Calculated the average relative to each of those plots. But let's say we want to convert that into something else useful. So for dandelions, we want to calculate the relative frequency of dandelions in each plot. So what we can do is go up here, and we want to insert a new column here because there's no room between dandelions and earthworms. It's column D to column E. Right-click, oops, Control-Z works in Excel as well. Right-click and press Insert. And all of a sudden now we've got a new column here. Now, because I'm going to be calculating multiple parameters that describe dandelions, what I want to do is create a heading for these multiple parameters. So you'll see both of these columns are going to describe that. There's this merge and center feature. Click that. What it does is it makes a big cell that covers both columns. So we're going to say, okay, all of these are dandelions. Copy, paste. This is the number of observations. And over here, this is going to be fraction in plot. Now, if you have a really long term, maybe you don't want to have a column, a spreadsheet wide to describe this one parameter. So let's shrink this down. There's another way to handle that. If you click on it, you can press wrap text, and it's going to wrap the text so that you can make a taller cell that's not obscenely long to describe that term that you've just put in there. It's still one cell, it's just taller. In fact, you can make it as tall or as short as you like. Okay. I'm just going to break here. Is this too basic for you guys? How many people think this is like really old hat? I'm going too slow. Right on target? Okay, good. I don't know, and I don't want to assume anything, so I don't want to be patronizing, but I don't want to be too fast.
Also, if you guys didn't know, he said Control Z, that's the undo.
Oh, that's another assumption. Control copy, Control C is copy, Control X is cut. The difference is in cut it goes away, in copy it just makes the copy, and then Control V is the paste. I'm going to say a snide comment here. As much plagiarism as we see sometimes, Control C, Control V seems to be the favorite feature. So don't plagiarize. I say it in every single talk that I give in a broad audience. Don't ever plagiarize. We know we'll catch you and you'll be in trouble, so don't do it. Okay, so Control C, Control V, Control X, Control V is copy and paste and cut and paste. So we want to do fraction in the plot. If you want to know what portion of the biodiversity of this 1 meter plot is dandelions, what do you need to know? Like what percent is in this plot? Well, I was going to make them talk and throw candy at them. Thanks, Ramses. Okay. Okay, here we go. No, we got to set a precedent. You get the good one. I need these up here. So you need to know the total number of observations in that plot. So let's go over here. Total organisms. So how are you going to do that? You've got to add them up, right? So there's 2 ways to do that. You could do cell 1 plus cell 2 plus cell 3 plus cell 4, or you could do the sum. I don't expect you to know and remember all of these formulas. If you want to do something and you sort of know what it does, if you go here to the formulas tab, there's a whole slew of different formulas that are available. These are the logical, like if than, greater than, less than, all kinds of crazy things there. There's text operators, there's date, there's math. If you go under here, more functions, statistical, engineering, all kinds of just really sweet formulas that will save you a lot of time, and you can look them up in there. But I know I want to do the sum. That's a pretty basic formula. So I want the sum of all the observations in this plot: dandelions, earthworms, clover, periwinkle, and honeybees. So it's sum, and this is another cool thing I always forget to show you guys. If you click on the formula here, it'll bring up the help for that formula. It'll tell you how does this formula work. So the argument, the numbers that you have to have in there, and they'll give you sort of a fake spreadsheet that you can cut and paste out of and just play with that data. So if you're not sure exactly how it should be in there, okay, that's fine. That'll be really useful. If you press this button right here, the little FX, it brings up a list of the variables that you can put in there. So if you're not comfortable just shorthanding it like I do normally, you can bring this up and it'll say, okay, what is the first number you want to add in your sum? What's the second one? And so on. So if we say, okay, the first number we want to use here, rather than typing 15, because depending on which plot you're working with, that number is going to change. What you want to do is use the cell. So this is a pointer or a reference command where, I don't care if it's Miles sitting in that chair or anybody, I want to know who's sitting in that chair. And so each time a new class comes in, it'll look in that chair. You want to think of cells like that. And so we're going to say, okay, I want to know if we take D3, number 2 is going to be F3, so earthworms. Number 3 is going to be that. And you'll see, because this argument allows you to add up as many numbers as you want, as soon as you add in the next number, it gives you a new cell to do that with. So we'll just do this, click, click. So we've got all 5 numbers, because there's 5 different things, dandelions, earthworms, clover, periwinkle, and honeybee, and we press OK. Hey, there's 64 organisms in this plot. So how are we going to take this sum and apply it to each of the plots? How are you going to do it?
Go to a row right-hand forward and then fold.
Right. Just like that. And so now we've got total for each of them. Oh, you want to go all the way down? So this is the average number of organisms in a plot. Right on. You want Hershey's? There you go. Charlie? Is that your name? Bob?
Bob.
Okay. Nice to meet you, Bob. So that's our total number of organisms in each plot and the average number of organisms in each plot. It's pretty neat, right? Because what it's doing is, say, sum, and it's adding up all of these. It color codes those cells for you to try to make it as easy to understand as possible. If I click on any one of these formulas and I go back up to this formula bar, so you want to think about this as like the address bar you would use when you're in the internet. If you click on that, all of these cells turn a different color, and it shows you where they are here. You can even change them a little bit, but we're not there yet. You'll learn something new every time, Ramses. The one thing that I will caution you is when you click up in the formula bar, your mouse serves a different function. If you click over here, whoops, if you're up in the formula bar, and let's say you're in here, and you click elsewhere, oh, it's not doing it right now. They must have fixed that. It'll replace that formula sometimes with the new cells, and so you have to make sure that you don't change it. If you want to get out of there without changing your formula, you press the escape key, and it backs out without making any changes. Okay, so we have total organisms. Our goal here, though, is to determine the fraction of those in the plot. So what we're going to do is do equals, and this is just simple math. The fraction of the plot is the number of individuals divided by the total, right? So we're going to say, okay, number of dandelions divided by total. Enter. We get a fraction. Are there questions up to this point? Do not be shy. Go ahead. So what we've taken here is the number of dandelions. So this whole thing here is dandelions. Observations in plot 1 is 15. And so I took this number divided by the total number. And depending on what you want to do, it'll be different, right? I'm trying to use a more generic example. But if you know the way to calculate a fraction of something is going to be the number of individuals divided by the total. If you were on your calculator and said 15 divided by 64, this does that for you. And then the nice thing is you'll be able to copy it down. Okay. You get to be encouraged for asking questions. So now we're going to copy that down. Okay. I'm going to do this quickly for the next few. Control Z. Insert. Insert. So we're going to move this stuff up so you can do Control X, Control V, X, V, X, V, X.
If you notice, the insert the column is always to the left. So
Mhm.
inserts to the left.
Yeah, it sort of inserts in that position and then like forces everything off to the side. So we're going to now take these terms. You can copy and paste text just as easily as you can numbers, so we're going to do that. And the formula, if you just take this whole column, copy and paste, oh, this brings up a really interesting thing. Why is it dividing by 0? It'll give you an error. It'll tell you when there's an issue with your formulas. Why is this spreadsheet angry that I'm trying Well, you know you can't divide by 0, but what's the problem? What's it trying to do? Think about what's happening in the cells that I copied. You see there's sort of the dancing outline. I copied from here to here. Why is it angry when it's here and it was happy when it was there? What did I tell you about moving formulas around? What's that? Right, it was this number divided by this number. And so it's measuring how many cells over was this number. When I go here, it's picking up this number, but it's trying to divide it by a number over here that doesn't exist. This is where having a little bit of knowledge about what it's doing is going to help you. So if we go back here to our original formula, and we look up here, this is the number that we want to have update for each different parameter we're working with. But N3 is the total number of organisms. Whether you're talking about dandelions, earthworms, clover, whatever, that total number for that plot is the same. So we want to fix that reference. So when it's pointing to this seat, so if I point to Miles right here, and then I move over here, and now it's pointing to George or to Rebecca, if I want to always go relative to Miles' seat, so when I move over here, I'm still pointing at you, and I'm coming over here, and I'm still pointing at you, there's a way to do that. If we go up to the formula bar, N3, we want the row to be able to change because we want to copy it down. But we want the column to stay the same. So what we're going to do is go in front of the N, put the $sign. The $sign means that that's a fixed reference. No matter where you move this formula around, it's always going to be that column. So I'm going to update this all the way down. And check this out. Control C, Control V. Now it works. Control V, Control V, Control V. You just learned something. That's the goal. Yeah. And so you can do it with rows, with columns, or both. So sometimes there's like one absolute value that you never want to change and always refer to. You just do $column, $cell, $row, and it'll fix that cell no matter where you move that formula around. But you'll notice that the first one is updating dynamically depending on where it is. So when you put that $sign in, it's not doing it for all of the cells, just the one that you put the $sign next to. Okay, so we played around with entering data and doing basic math with it. Are there questions up to this point? I'm not going to do it because I'm taking a lot longer than I think I thought I was, which is typical for me. But you can take this calculated value and then use it as an input for another formula, and then another formula, and another formula. And so you can have all the different intermediate steps in a very long manipulation be dynamically updated. So if I go back here and I use, if I change the observations here, what you're going to see is, let me just change that to say 20. Watch all the cells that change. You see all of that stuff updated at the same time? That's because by changing the number of dandelions in that plot, it changes a few things. It changes the total number of individuals, because each of these fractions relies on the total number of individuals. Those numbers change. Because the average incorporates every value in the plot, or in the spreadsheet, those need to update as well. Okay? And so, if I go in here, oh no, that was 69. Boom, all of the things update in real time, and they're all changing relative to each other. This is great for basically any science class you take. Physics, this is a lifesaver. Chemistry, a lifesaver. Genetics, a lifesaver. I'm going to open up another spreadsheet real quick here just to show you an example. Analytical chemistry had some homework the other day. Some of you are in there, right? You've got 2 pKa's, right, for this, is that carbonic acid? And it has two ionizable groups. At very low pHs, both of the acids are protonated, and then as you increase the pH, you start to deprotonate one ionizable group and then the other one. And so there's three possible species of ions or molecules in this solution. Both of the protonated at both sites, and then as that concentration decreases, the concentration of the species that has only one protonated increases. And then as you increase the pH further, you abstract another proton, and they're both deprotonated, so a -2 charge. This was the homework, and the formulas, or the known values that you were given, were the two pKa's, so the first group and the second group, and these equations. So the pH relates to the concentration of hydrogen ions in the solution using this equation. And the relative concentrations of each of these, this is Henderson-Hasselbalch, if I'm remembering correctly. And so it describes how each of these are related. So this is written out in words. But concentration of hydrogen, you can calculate from the pH. So if you change the pH, and this is something you would do experimentally, so an independent variable, this is Alison Martin's data, so I'll give her credit. What she did was fill in all these pHs on the side, and then the first thing she's going to do is calculate, well, what's the hydrogen ion concentration from at a given pH? And so she just uses this formula where it says 10 raised to the negative pH. Okay, that gives you the concentration of hydrogen ions. And you'll see that A3 refers to the pH cell. So this is the thing you're manipulating, and then the response is the concentration of hydrogen ions. That's a value you need to solve this equation. And so she's done that really well. All you have to do is do it once, copy it down. Next, well, what's the concentration of the conjugate acid? Right? So you've got the formula here that relates hydrogen ion concentration. So if we click on that, this is the, I'm going to just step out here. This is the hydrogen ion concentration. We're looking in this cell, but it's referring to this one. So here's B3, here in this formula. And then it's looking at the two pKa. So here's pKa1. You need that. Why do you think she has dollar signs in there? For both row and column. Right, Geraldo, you get candy. All right. So these values are sort of physical properties of that acid. And so they're not going to change regardless of whether you change the pH. And you want to always refer back to those. And so she's locked that reference for pKa1 and pKa2, but is dynamically changing the hydrogen ion concentration as she moves through different pHs. And so what you're doing here is we move up from very low pH to very high pH. And what you can see is the concentration of the carbonic acid with two protons still attached to it is high here, and then all of a sudden it falls off, which makes sense. When you take acids to a more basic pH, you pull off a proton, and so the concentration of that species falls. The goal here is not to teach you chemistry, but to show you how Excel can simplify those calculations for you really easily. And so now we're seeing that. And she's done the same thing for the species with just one proton removed and both protons removed. It's how you apply that formula. Knowing the formula isn't important. Knowing how to enter a formula is. I guarantee you what she did was entered all these pHs and entered the first row of formulas. And then what she did was go like this. You can copy multiple formulas down and do the entire sheet in one go. And so she probably did this, pulled it all the way down, and voila, it fills in.
In fact, I know that's how she did it because she told me.
I'm sure. Hey, Phil. In a matter of, I'll bet you it didn't take her more than 10 minutes to make this entire spreadsheet and solve every individual value that she needed for this homework assignment. Making plots is the next thing I want to tell you about. Actually, there's one other thing that I do want to show you. Conditional formatting. You know, my roulette spreadsheet, I had things changing colors in response to the values. If you want to do that for any of your spreadsheets, you can highlight the data that you want to format. Over here, there's conditional formatting. I like these color scales. So if you click on that, what it's going to do is give it a color that is proportional to the value that's in there. If you manually go in and type a different number in here, it'll update in real time. So at a real quick glance, it lets you look at it and say, oh, this is a high concentration, this is a low concentration. An example of how I use that on a regular basis. So I teach genetics, I stripped all the names out of here. But this is a giant spreadsheet of every grade that everybody got in genetics this past fall. A quick glance, if I say, okay, I'm interested in student Z, I can look across and say, oh, they did really well here, not so hot over here, just by looking at the color of their grade. Because sometimes the way you present data, the number doesn't mean as much as sort of its relative abundance compared to other things. Another thing you'll notice is I've got these little drop downs here. This is a feature that you're going to find very useful as well. If you want to sort things, let's say I want to know who did the best on their exams. Well, student A did. They got 99.6 out of all the exams in genetics. If you do that, you're a rock star. I applaud you. You should all aspire to do that. But I can know, okay, student A was the best. Student AW, these are not initials. This is just A through Z and then AA, AB. And so student AW was 38%. And so that was the range of scores. All of this stuff updates in real time and allows me to do plots like this, histograms for different grades, all that jazz. I did not give you this spreadsheet in that shared folder for obvious reasons. I don't need anybody trying to back calculate who got what. So how do I get to be able to sort data? Let's go back to our example over here. If we have these different observations and we want to sort, what you want to do is highlight the range of values you're interested in sorting. You do not want to include the average usually. So make sure you're not including that. If you click on the Data tab and Filter, all of a sudden you'll see the header, the column headers will show these little drop downs. And so let's say I want to do based on the number of dandelions, largest to smallest. Oh, it sucked in average there. Insert. I need to space the average away from that a little bit. We're going to turn on filter again. If I want to filter based on these largest to smallest, what you'll see is now we've got 20 at the top, 3 at the bottom, so I can get a real quick look at which plots had the most dandelions and which had the fewest. When you set up a range to filter, what it does is it keeps the whole column together. So when you move that one column, it moves all the other parameters with it accordingly. So you're not separating plot 1 and plot 2 as you go through it. That's a important thing to realize. Somebody, I can't remember who it was, I think one of my research students came up to me the other day asking how to do that. So that's how you do it. Questions at this point? I know I'm running a little late. Sheets here. I want to make sure that I've got all these. Okay, I just did conditional formatting. Pasting values and not results. This is something people get hung up in all the time. You want to take these numbers. They're really interesting to you and you want to put them somewhere else. So you do Control C, Control V, it's divide by 0. How do you get around that? You just wanted the numbers. You didn't want the formula. See this little thing down here? You click that and it gives you a whole bunch of different pasting options. The one that I find most useful is this paste values. If I click that, now this is a hard value that will never change. It's not a formula. It's a solid value. I just saw a junior with a light bulb going off on his head.
I needed
Now you know.
I'm always thinking manually after.
Let's say instead of having them vertical like that, you want to change a column to a row. Right click, and here's all the paste options. This one, the transpose, is kind of cool. And you say paste values. Oops. I'm going to copy that again. This transpose feature changes the numbers from a column to a row. A graduate student is now having a light bulb go off. So these sort of things are going to make your life a lot easier rather than retyping all that stuff. Use the tools that are already in there. All right, I'm saving lives by showing how to paste. This is great. A very simple application of this was in my molecular genetics lab. We have a multi-channel pipetter and 96 well plates. So we have 96 samples arrayed in an 8x12 plate. And you have a pipetter that's going to take 8 samples at a go. And then I pipetted that into a gel. Well, that should be pretty easy, right? It keeps you organized. Well, the issue is the pipetter went in to a set of wells on the gel, but it skipped a well. And then so you had another set of samples interlaced with it. And keeping track of like what was in which well when I looked at the gel was a little bit challenging. And so I said, I'm going to make an Excel sheet to solve that problem. So here's the 96 well plate with 8 wells in this direction and 12 in this direction. I was able to take 8 wells at a time, but how did that get moved into the gel? I can type in what I know is in each of these samples, so that it's a sample, it has a name that I've given it. But because these samples are interlaced, here's the purple row. You got this sample, this sample, this sample, and this sample. These names here refer back to their position in the 96 well plate. This is a tool that I can use over and over and over again. All I have to do is delete these values up here and say, okay, now I want to do sample A, sample B, sample C, D, E, and F. And what you're going to see is these are updating down here. Here's A, B, C, D, and E. And then when I look back at my gel and I see bands over there, I can just count the number of rows and immediately know what's in there. Excel can do all kinds of stuff like that. The thing that I don't want to forget to show you, and I can't believe it's already 8 o'clock, is how to make a chart. So everybody wants to make graphs and charts out of their data. It's an important way to communicate your science to your reader. So if I want to make a basic sort of histogram, so bar graph, a lot of people will say, okay, highlight all your data and then click Excel and hope that it makes the graph that you want it to. Sometimes it does, very often it does not. And that's because you're sort of relying on Excel to figure out what you want. What we want to do is insert a graph. And so we're going to do so we're going to do this, and I'm just going to do this sort of a clustered column chart. It's totally blank. There's not a bit of data shown here yet. So if you click on that, there's this select data, or you can right click as well and select data. This is your friend. You always want to use this to enter your data rather than hoping that Excel picks the right variables and puts them in the right spot. I remember being a freshman in college and we were doing enzyme kinetics in my very first intro to cell and molecular lab. And we were like rapidly like moving columns around and clicking Excel and hoping that it made the right graph, and eventually it did. And that was really not a smart way to go about it. What you want to do is go here and just say, okay, I'm going to add a data series. So the series name, rather than typing that in, I'm going to click dandelion up here. That means if I were just to change what I call dandelions, I want to change it to the scientific name. It's going to automatically update everything throughout the rest of the chart. The values that I want to insert here, you highlight that, you say equals. Let's do observations. Click. See how it's adding all those observations in here? Okay. The horizontal categories. So we have plots as our categories. We're going to edit this, and we're going to highlight that. Okay. And so now it's putting anything that's in whatever we're labeling each of our sets of data. This is our independent variable. It's putting it over here in this horizontal axis categories. Now we want to do not just dandelions, but the other stuff too. Let's add a category. Earthworms. Okay, so this is going to be equal to earthworms. This is going to be equals to the number of earthworms. Click. It's throwing earthworms in there. You can see there's none in plot 5, so we don't see a bar there. Great. Let's add them all. So we're going to here, series name, do clover. Okay. Add series name, periwinkle. Any questions up to this point? No?
Well, while you're doing that,
Yep.
it's putting them after you ordered them. So if you reorder using the drop down, does it reorder the table?
Check it out. So we're going to do plot, smallest to largest.
And
Yes, immediately. Now, this graph is not, or this chart is not good. Why? Labels, everybody, that's great. I'm sure everybody's lost points because of that. So click on that. What we want to do is go back here to, under chart tools to design, add chart element. You add whatever element you want. So we need axis titles, primary horizontal axis. And so this is plot. Oops. Okay, add chart element, axis titles, vertical. And so this is going to be, you can just go in here and highlight it. Okay, like that. You need a legend. What are all of these different colors? So click on that, design, add chart element. You need a legend. So here's our legend. We want to place it somewhere, so it has it over here. You can drag that around inside the chart, put it up here. You can grab the chart and change how big it is. There's lots of other sort of clever formatting things which are not as important, but you can go over these and it'll give you all kinds of different chart styles. I tend to like this one. I got rid of the legend for some reason. And there you go. You've got a nice, easy to see. Nate.
So say you wanted to make like 10 graphs, you had to make like 10 graphs like this,
Mm-hmm.
or like a separate one for dandelion or whatever.
Mm-hmm.
Is there a way to standardize the format so that it shows up the same color, the same?
So what I would do is, and there's probably multiple ways to do this, but you could say copy and paste and select different data for this set. Okay? And so it will look identical except that you input different data. Okay?
I know how to do this, but for something like this, where you would want the numbers displayed over the bars because there's so many different categories, how do you do that?
In terms of,
So like just put the number in the n equals.
so if I was to do that,
Yeah.
so you can see if I select a given series, so I want the blue or the gray or the red. So let's do that. You can right click on it and add data labels. So we do this. Boom. You've got numbers right above it so you can see how many individuals are on each of those. You can do it for all of them. You can do it for a handful of them. This helps you to draw your reader's attention to the data that you want to show. If you'll see that I highlighted that, or if I clicked on this, it highlighted all of them. If I want to highlight just this specific example in plot number 1, you click once, and then you click again, and it'll pull just that one out. You can format that data point to make it extra fancy. So fill, gradient fill. I mean, I'm just going to be ridiculous here for a second and say for this, it's going to be red. And you can see now it's like a real fancy bar because that's what's the most important thing to you. There's so many options. So this is one type of chart you might make. The other major type of chart you're going to make is a scatter plot, where you put a trend line relating two different variables. So let's insert that. Before I move on, are there other questions about charts? Is this useful? Yes? Okay, good. I hope so. Insert. We're going to do a scatter plot, and let me do it down here. You can have the line in there or just the scatter. We're just going to do the scatter. Again, it comes up blank. I like starting from that blank slate and then adding my data rather than, like I said, hoping Excel picks the right thing for me. So we're going to select the data. Here we're going to add, and you'll see that it's a little bit different. This is the series name. And so we're going to give this a name of Clover versus Honeybee. Whoops, we have to edit that data. So on the x-axis, let's say we want to see, if we look at plots with different amounts of clover, how do honeybee prevalences vary with clover? I happen to know that honeybees like clover, but let's see if the data bears out that hypothesis. So on the x-axis, that's going to be your independent variable. And so in this case, it'll be clover. Okay. And then the response variable, the dependent variable is going on the y, and let's say that that's honeybees. Like that. And we say OK. And OK. So right here, this is clover on the x-axis. Of course, we want to add our axis labels. Primary horizontal is going to be clover plants. And our vertical axis is going to be bees. And you can see with low prevalence of clover, there's only one bee. With a little bit more, you have two, and with even more, you have three. Well, that's kind of cool. But how are they related? Let's make a formula. Click on those data, and what you'll see is it highlights the data that are being used to generate that chart. If you missed a sample, let's say you wanted to throw the averages in there as well. Get this out of the way. Click. Okay, so pulled that down. And so we've got the average value, which is right in here. Typically, you're not going to want to do that. You don't want to insert your average as something on this sort of a plot. But we're going to click those data points, right-click, and add a trend line. And there's a couple different sort of trend lines you can insert. I'm just going to do the most basic, which is a linear regression. So you say, I'm going to do a linear. Let's display the equation and the r-squared value on the plot. Done. It shows you this trend line, gives you this equation to solve this. In chemistry and physics and even genetics, you're going to be generating standard curve data and then taking an unknown and plugging it into that standard curve to figure out what the concentration is. This is how I do it all the time. You put it into this, you generate the standard curve, you fit a trend line to it, get the equation, and then you can solve for any of those variables. Question?
Yeah, just so you guys know, the intro to some molecular students, that equation there ends up being here as well. Okay, so you would input your absorbance, in the X, and it would give you the concentration of the Y. That's the same formula if you make a standard curve using this stuff.
Alrighty, so what I'll do is I'll take questions for the next couple minutes if you have them. Excel does so much more even than this. Anybody? I'm going to show you one that Ramses and I use. It's pretty useful, I think. So we're doing this experiment in the lab where we need to put a specific molarity of RNA into a reaction. But when you take the value of this RNA, it's the value out of basically a spectrophotometer. And so how do you convert that to molarity? You need the molecular weight. It's a So for each of these different RNAs, we know the molecular weight. And so this is a known. When we take our assay concentration, figures out the dilution, the units, the stock concentration, takes the molecular weight, converts it to molarity. And then in terms of practical use, we need to know how many microliters of this we have to add to that to standardize that concentration. This Excel spreadsheet does all of that for us. And so this is in that shared folder, which using your email addresses, I'll share that. And you can go in there and feel free to like tear it apart and put it back together, use these formulas to solve whatever problem it is that you have. The last one I'll show you, for those of you that are getting ready for genetics, where is it here? I'll open. There we go. When I make your homework questions, I have Excel sheets that make these questions for me. So there's new numbers each time I make a question. And so things like the binomial probability questions. Okay, if I expect a 1-to-1-to-1, and I say, okay, I want this many trials, it gives me all of those answers. So when I'm generating the solution, all I do is enter in the numbers and boom, it does all of those calculations for me. I will not share this with you. You have to build one for yourself when you take genetics. But almost all of the quantitative type questions that I've ever given you are made using my homemade Excel spreadsheet. This would be a very valuable one, so I'm not sharing it with you. Not because I don't want you to see how I've done it, but rather because this is an opportunity for you to take questions, and I know a lot of you are sort of rising to genetics next year. Understand the formula that I give you, write an Excel equivalent to it, and solve your problems perfectly every single time. There's no rounding errors. There's no sort of transposing numbers accidentally on that piece of scrap sheet that you paper that you have. It's just a great way to be as precise and organized as possible. So that's that. Questions? I know I ran long. I apologize for that, but I think this is useful. Here's the thing, it saves me time. And I want you to think, that's an interesting question, George. He asked, how do I find time to make this stuff? Making it takes maybe 20, 30%, 50% longer, but when you do your homework and you get the wrong answer, and you have to go back and recalculate the whole thing again, oh crap, I did it wrong again. Okay, I got to go back again. With this, all you do is you tweak one little variable. You change the way the cell refers, or you add one thing to your equation, and all the calculations are done immediately. So while it might seem cumbersome to make it at the front end, it saves you tons of time on the back end. And so I find it totally worth my time to do it. And once I've made it once, I can reuse it and adapt it any way I want. Like this one that Ramses and I are using in the lab, we go in and we tweak it each time we do it based on the variables that we want to use in the lab that time. And there's no cumbersome calculations. You type in what comes out of the spec, and it tells you how many microliters you need to move from one tube to the next. And so it saves you time. And I think that time invested is totally worth it. Oh, cancel.
I don't know, you never teach you how to really use it.
All of this will be on YouTube, so you can have fun with that. All right. Where is my... There we go.